Okay, it is 2 o'clock. I'm gonna go ahead and get started. So this is a fully asynchronous online course, but I do think it's nice to sometimes just have an option to join me for a little lecture real quick. So on Mondays, either before or after office hours, I'll be lecturing on the chapter. So this week we're doing chapter 1, so I'll have this kind of quick chapter video. This one will be a little bit on the shorter side because the chapter 1 is pretty straightforward. Some of the others, they might be a little bit longer, but none of them will really be over I don't expect more than like 40 minutes for on the long side. Generally, the soft chalks, the lessons kind of walk you through that stuff. So if you don't need to watch the videos, that's up to you. But I do think that it's usually helpful to get to hear me explain things and highlight different concepts. And like I said, these are, I'll have the times for when these are going to be recorded, posted in Teams. So if you want to join live, that way you can ask questions, you absolutely are welcome to do that. All right, I'm going to turn off my camera while I do this, and we're going to dive right in. So for chapter 1, we're just getting into our introduction to the study of life. So just kind of introducing some basic concepts, talking about the basic characteristics of life and how do we define life as biologists. That's what we're supposed to do. It's the study of life. So how do we do that? So we have to start off with just some basic things of, well, what does it mean to be alive? What is life? So all life is made of cells. All organisms are made of cells, whether it's unicellular or like humans, where we have 6 trillion cells about on making us up. Everything is made from a cell, and we'll get into some ideas of cell theory. These ideas that cells come from one another. They don't just, there's no spontaneous generation. For a while, that's how people thought life was made, was just spontaneously generated out of the air. When you see just, all of a sudden maggots crawling along some decaying material, you would think, oh, those just came out of nowhere. But now we know, oh no, there's microscopic eggs and they grow into the maggots. And cells do this. Cells make up everything living around you. All life has DNA. All organisms have DNA. We're going to learn more about DNA. We have several chapters dedicated to it, but this is what carries the genetic information. It is what codes for the proteins that make up your structure, a lot of your functions in your body. And so DNA is that essential kind of building block for life that we're required to have. And again, the DNA is used for protein production. We're going to see as we get into our macromolecule chapters that proteins do a lot of the work in the body. My joke is always, if somebody asks you what is the function of this molecule, or what kind of molecule is this, just guess that it's a protein. Proteins do everything. So now we get into some more of the specific characteristics of life. Depending on what resource you use, they're going to talk about 5 characteristics. They might talk about 6 characteristics. They might combine these and add a different one. So you really have to just kind of take this with a grain of salt, that from this perspective that we're learning, these are the characteristics that they share. If you go, like I said, and look at something else, they might have some slightly different characteristics, but it all comes back to these same kind of concepts. This first one is that life is organized. Life has this basic kind of structure that puts it together. You see kind of the smallest down here is the atom. Something that is just an atom, something like talking about a carbon atom, an element, an atom of hydrogen, an atom of helium. Those things on their own do not have any properties of life. So an atom is not technically a living thing. And when atoms come together, they make molecules, and that's still not a living thing. A molecule would be like, they're showing DNA. A water molecule, H2O, that's, several atoms coming together to make a molecule. They have a little more complex function. We really highlight the more organized you are, the more complex your function is. But still at the molecular level, the organelle level, you aren't living. You don't have the properties of a living organism. Organelles, we'll see, are little organs. They're organs that are inside of cells. And a cell is the basic unit of life, the fundamental unit of life. Nothing smaller than a cell can really exist on its own as life. So it needs to be that organized at least. And a cell is essentially a boundary that has had a collection of organelles. You have multiple organelles all working together within this boundary towards a common function. And so you can have a bacterial cell, a unicellular organism that is fully functional, fully living, right? And then as you get more and more complex, organization, you get more complex structures. So depending on where you go in biology, you'll kind of study all the way up to the biosphere. This class, Bio 111 specifically, we're going to stop at the cell. We're going to get kind of through DNA, through cell structures and functions. And then once you get into Bio 112, that's when you'll start going into tissues, organs, organisms, studying ecology, and some of these other concepts. And so, yeah, just depending on where you go, you'll kind of have somewhere along this line, this chain of being. All right. So this organization, just kind of staying on this organization for another minute. I hinted at this or talked about it directly, that complex structure, complex function. The more organized you get, the more complex your function is going to be. And when we call this emergent properties, these properties that arise to emerge or to arise as you start to get more organized. So this example is showing just one brain cell. One neuron on its own doesn't really have, it can conduct an electrical signal, sure, but it can't really give you consciousness. But once you start to have trillions of brain cells interacting with each other, it starts to give this rise of consciousness. You start to get these complex interactions that create our personalities, our ability to think and predict and reflect, all of those different things. And so we get these more complex functions. The sum of the whole is greater than its parts. And so this is, again, something that you'll see as you go, especially if you take classes like anatomy and physiology, you'll see how things kind of get put together to give organs their complex function that they have. You can kind of think about this, with the digestive system. One digestive organ on its own, okay, that does a little bit, but it takes the entire system to be able to fully process all of our nutrients and absorb those. Life requires energy. This is a basic component, a basic characteristic of life. All life is going to have to require it in some capacity. There are three essential components to this chain of energy. You have your primary producers. These are things like plants and algae that go through photosynthesis. They get their energy from the sun. Consumers are going to eat your producers. So insects might eat plants. You have carnivores, omnivores that also will eat some producers. They'll eat plants, but they'll also eat some of your lower consumers, lower down on the food chain. And that is that transferring of energy. Right? So as producers make their energy, they get their energy from the sun. That is the main source of energy in our universe, is in our galaxy, but across the other galaxies as well. The main source of energy is the sun, putting off all of this radiant heat, this radiant light energy. And so when we get into photosynthesis chapters, we'll talk about how producers can harness light to turn it into physical sugar. They'll trap carbon dioxide from the air, light from the sun, they turn it into a physical sugar, and that's what allows them to grow their stems and their leaves and their fruits and all of those different things. Consumers eat those, and then something potentially eats the consumer, or something breaks it down, like a decomposer. So a decomposer are specific types of consumers that get their nutrients from dead and organic waste, from dying dead organisms. So they don't necessarily go out and kill mushrooms. Well, mushrooms can be toxic and very poisonous and dangerous. When you see them coming out of the ground or coming out of an organism, typically it's that they're decomposing that organism. They didn't actually cause the death. They're just breaking it down. They're part of that cycle to reabsorb nutrients back into the soil. And you can see that's where that goes. And then that soil, those nutrients are then provided to the producers. So it's this circle of life. Should be pretty familiar with this. One thing I want to highlight, though, is this heat loss that happens across each of these energy transformations. That's what we consider this, going from producer to consumer to decomposer and around. These are energetic transformations that are happening. And each time you're losing a little bit of heat. And it's about 10% or so of heat is lost with each reaction, which is kind of why as you get larger as an organism, you have to eat more and more because these smaller organisms, if you think about trying to just survive on something like shrimp, if all you're going to eat is shrimp, you have to eat a lot of shrimp compared to something like steak. Steak has a lot more energy available to it, and so less heat is lost in those reactions. And so we'll get really specific into these different energy reactions later in chapters 4, 5, and 6 as we go through this. Life requires homeostasis. This is an essential thing, but we're constantly come back to, if you're studying biology, you're going to always talk about homeostasis. This applies both to individual organisms and to ecosystems, to our biosphere, to the Earth as a whole. It has to be in homeostasis. When we experience these extreme weather events, the flooding that we've been experiencing lately, especially if you're living in Raleigh, we've been having some wild flooding happening downtown. That's homeostasis. that is homeostasis happening. That is the water table trying to find an equilibrium. As the ice caps melt and the water table starts to rise, groundwater gets filled up. And so when it rains, there's no space for the ground to absorb that extra rainwater. So it just has to sit on top of the road surface. So yes, construction has something to do with that, but also the water table thing. Sorry, that's just been very prescient in my mind the last week or so. But that is homeostasis, right? Homeostasis, we're showing this example of looking at a thermostat. A temperature regulation is always the classic example for homeostasis. Blood sugar regulation is another classic homeostatic mechanism. But almost everything that we go through is regulated through homeostasis. Blood pressure, your sleep rhythms, your hormone rhythms, they're all kind of reaching this homeostatic balance. And what's trying to happen is that the body, or life rather, is trying to get to this set point. There is kind of a set point, right? Like if you think of you're setting your thermostat, you put it at the temperature that you want it to stay, 66 or 70, whatever it may be here. And that's the set point. And so when it starts to deviate from that, either the heater turns on or the heater turns off, the AC turns on to return it to that set point. The thing with life, though, with an organic organism, with a machine, it can get to that point, but it'll start to deviate. Same thing with us. We're never really staying at that set point. It's always going to deviate a little bit. And so our bodies are constantly working to reach homeostasis. What this means is that if you are sick, if you are recovering from surgery or injury, anything like that, then your body is having to dedicate extra energy to try to maintain homeostasis. That's why you're supposed to rest, hydrate, get proper nutrition when you're recovering from something, because your body's having to work extra hard to try to maintain that homeostatic balance. And especially for us living in the South with the heat that we get to experience and the humidity that we experience, if you're already sick and then you're putting yourself in extreme temperatures, your body is really going to struggle to maintain homeostasis, and then you could pass out, you could get sick, dangerous things could start to happen. So this goes into internal chemistry as well. So like I talked a little bit about temperature. This one is looking at hydration as well. So your fluid balance, your water balance, your electrolyte balance, those signals that you get that, oh, I'm hungry, oh, I'm thirsty. Those are all your body trying to maintain homeostasis, trying to maintain that proper balance of sugar, salt, water, nutrients, whatever it may be. Okay, these next three, they kind of go together: reproduction, growth, and development. These are the kind of three characteristics that we kind of pile together, and then we'll separate them out. We'll talk about what each one is. Reproduction, that one tends to be a little straightforward, right? Things reproduce. You have to create offspring to be able to sustain life and propagate life. And so life reproduces. It can be different forms of reproduction, though. Asexual reproduction, where you have one parent creating identical offspring. You can see here in this image the strawberry plant. A strawberry plant, you have the parent plant, and then it sends out those runners, and each one of those is an identical clone to its parent. And so that is an asexual reproduction. Bacterial cells, they go through asexual reproduction. This is really successful in unchanging environments. That's something you want to make sure you're highlighting, is that asexual reproduction is successful for unchanging environments. It's successful if you have really quick reproductive rates like bacteria do, where even if they do get put into changing environments, they can reproduce every 20 minutes or so, and they're able to adapt really quickly. But with asexual reproduction, because you have you're limiting the amount of genetic variability, you're limiting the amount of mutations that are accumulating, and mutations are adaptations. Those are what adapt you to your environment. And so if you have an unchanging environment where there's not a lot of ecological distress, no sways in temperatures or fires, any kind of ecological disasters, then asexual reproduction can be very successful. Mostly because you get really suited to that environment. If you are able to be successful and reproduce through asexual reproduction, you're probably very specialized to what it is, that space that you live in. Sexual reproduction, though, that is when you have two parents involved, and the offspring are genetically different from the parent, right? They share traits, they have this blending of the genetic traits, but they are still different from their parent and different from any kind of siblings that they might have, excluding identical twins. And so sexual reproduction is really helpful in changing environments. It increases the odds that you're going to have some kind of adaptation that accumulates that makes you more successful. And we talk about reproduction, we talk about, in this case, reproduction, fitness. We talk about that in terms of evolution. That means the ability to reproduce. And it means the ability to make more offspring. That is kind of how, from a biological standpoint, we determine natural fitness. Really, though, it's just kind of about this idea that, well, when you have sexual reproduction, you're going to have this blending of genes, and some of them might accumulate traits, right? You might kind of double down on certain traits, skin tone or height, metabolic processes, right? Those kind of things. Or it could start to deviate. You could start to then pick up a recessive trait, someone that was not showing in either parent, but now is present in one of the offspring that actually inhibits their fitness. And so it kind of is this, slight gamble that you play with sexual reproduction, but it does overall provide you better odds against changing environments, which we very much live in. So now let's start talking about growth and development, because these are two different processes. Growth is the increase in size, right? Size by cell division. So that is just something getting larger. A little fun fact though, when you go to like build your muscles, when you go to make your biceps bigger, you're not building more muscle, you are making those muscle cells bigger. They are actually increasing, not by division, but just by increasing in their size, the amount of protein content within the cell. When you start to actually increase the number of cells, that's a lot of times when you start to get things like tumors. So that is that hypertrophy versus hyperplasia kind of thing. Hyperplasia is a tumor that's actually increasing the number of cells, whereas growth, again, cell division, especially if you think about like when we're going through embryonic and fetal development. For that fetal development, after we kind of develop our initial structures, we're just growing in size, right? Most of the pregnancy is spent in just kind of developing and increasing the size of the fetus until it's ready for birth. Development refers to changes in physiology, changes in your ability to do something. And so this is kind of like during that embryonic development. So the first 8 weeks or so of pregnancy, we're going through a lot of physiological changes, creating those rudimentary structures of the nervous system, the cardiovascular system, the digestive system, and then through the fetal development from weeks 9 to 40 or so, those kind of that fetal development, we're just increasing a lot of size, a lot of growth that's happening. Right after birth, there's a lot of growth that tends to happen until we hit puberty, and then we start to change our physiology. We start to get these different hormonal signals that then start to change our actual function. We're now able to produce, females are able to ovulate, males producing sperm, and so we're able to reproduce. And then that doesn't stop. A lot of people tend to think, oh, you go through puberty once and then it's done, and that is very much not true. There are multiple points throughout your life where you're going to go through developmental changes, where your hormone levels are going to start to shift, and your body's change. Your body's going to go through different physiological processes. Males, females, we all experience it. And so that is that difference between growth and development that we just want to make sure we're seeing. Okay, last one here, life evolves. Life is going to change. That is just kind of one of those aspects of it. When you introduce these unknowns, right? We live in these uncontrolled environments. As much as humans like to try to control our environment, we cannot be perfect at that. Diseases, natural disasters, all these kind of things can arise. And so when you have that change that's being introduced, life is either going to end or it's going to adapt and find a way. And typically it finds a way. A lot of people like to kind of be doom and gloom about us ending the world and humans destroying the world. And let me go ahead and be if you haven't heard it before, we are not going to destroy the world. We might destroy humans. We might make it uninhabitable for us and for a lot of other organisms. But life will evolve and life will change and find a way. And hopefully humans can keep up with that. So here you see the generation of antibiotic resistance being presented in bacteria. This is a really big one that is very prescient in medicine and public health, right? You have colony of bacteria, and then let's say that there is a mutation that occurs, and the mutation that occurring, this is going to be antibiotic resistance. And so what happens is that these all bacteria, they are all existing just fine on your skin in this cut, no big deal. But then you go to the doctor, right? Let's say you decide to go to the doctor, and it's like, oh, I think it's getting worse, so I'm going to go to the doctor, and I'm going to ask them to give me some antibiotics. Well, when that doctor gives you antibiotics, it's going to kill off all the green good bacteria, or all the green non-resistant bacteria, and that's going to allow this resistant red bacterium to have all that access to the food and the space and the resources so it can propagate. Right? Versus, well, if you didn't do antibiotics, it doesn't mean that you're going to get rid of the bacteria. You're just going to not get rid of the good bacteria, so the antibiotic resistant can't propagate. This is a kind of an interesting little concept, so you might have to go back to it a couple of times, and we'll do another slide on this in just a moment. But this is one of those things that happens if you, next time you go to the doctor, listen to them or read the instructions, and they'll tell you if your infection does not get better, if your infection gets worse, if your conditions worsen after a certain number of days, contact your physician. And they want you to do that because a lot of times when you go to the doctor, they don't have time to sequence what exactly kind of bacteria this is. They're just going to give you some general antibiotic and assume that it's going to work. And when it doesn't work, that's when they know, oh, you have a resistant strain. You need to come back in. We need to test this specific bacterial strain and give you a specific antibiotic for that. That's what happens with antibiotic resistant bacteria. So this is just showing another example of evolution. So rather than something like developing antibiotic resistance, this is a seahorse that's really fit perfectly to its environment, right? It's this little pygmy seahorse. It blends in perfectly with the coral reef. You can't say it's going to be the strongest or the fastest, so it has developed these really great hiding mechanisms. It's a DNA, everything that you're seeing, the bumps, the colors, those are all proteins that come from its DNA. So it's the parents of this particular one were able to pass down those specific genes that gave it this coloring, that gave it those reactions, so that when you see a predator, you hide, you don't run, because when you run, you make movement and then something can catch you. If you're a camouflage is your defense, then you want to stay still. And so all of those things, all of those traits are coded in its DNA, how its nervous system is going to respond to seeing something out the corner of your eye. Some people jump really big, some people barely react. That's your DNA coding your nerves for how they send signals. And so all of those little changes are what accumulate in evolution to create an organism that is suited for its environment. So again, this is just going back to that resistant strain. I guess I shouldn't have gone too in-depth into it, but you can look at this slide again just to kind of reiterate that idea that, okay, so things can evolve. You have just this random change over time. As bacteria reproduce, it's just a numbers game. You're going to have a mutation. It's going to happen as much as we try to limit mutations in our DNA replications. It just happens anyway. And so over time, you're going to get this mutation. And then, yeah, it just depends. If you give them antibiotics, well, that's going to allow the antibiotic resistant ones to grow. And if you don't give them the antibiotics, you're going to have these kind of they're green. And so I don't want to say they're beneficial because that is true. That does happen. We do have beneficial bacteria. But in this one, we're just kind of seeing that the green bacteria take up all of the resources, so the antibiotic resistant one can't really replicate. Okay, so jumping out of the characteristics of life, that was kind of our last characteristic of life, that life evolves thing. I'm getting into this next section now, the section 1.2, looking at how we organize life. I'm very actually excited about this chapter. I haven't gotten a chance to finish a book yet, but I picked up a book called Every Living Thing, and it's all about the history of taxonomy and the history of how biologists organize life, because wouldn't you know it, it is political. Back in the 1800s, we used the Linnaean taxonomical system. Linnaeus, Charles Linnaeus, I think, was a particular. That was the way that he organized, chose to organize life. That is the system that we still use. That's what you're seeing here, where we have the kingdom, phylum, class, genus, order, species, that kind of organization in biology. That's the Linnaean classification system. And so taxonomy is not taxidermy. Taxonomy is the process of kind of classifying and organizing life. And the largest organization, the kind of most overarching organization, is called a domain. So you have the bacterial domain, you have the archaea domain, and then you have the eukarya domain. So three domains: bacteria, archaea, and eukarya. And then within each of these domains, you have more and more specific organisms. So like within eukarya, we humans are part of eukarya. So that includes animals, which are humans, fungi, like mushrooms, and then plants, trees, ferns, all those kind of things. And so we are all eukaryotic organisms. You have these archaea organisms. These are some, again, single-celled kind of organisms. We're not going to get too much in here. You'll get more into these when you do 112. And then bacteria, again, that'll be more 112, microbiology kind of stuff. But you can see that each of these has these kind of branching lines that as you go down, they start to come together. And then down here at the bottom, it talks about the common ancestor of all life. Sorry, just getting some water. So this is something that's starting to be a little more controversial, just because a lot of modern scientists are struggle with the idea that as big as life is, and as big as the Earth is, and as the ability for life to kind of inhabit all biomes within the Earth, that there would be one ancestor that then spread to the rest of the world. So usually what you see now are talking about multiple places where life began to arise. And again, we're talking single-celled organisms. We're talking very, very simple single-celled organisms that rose from the ooze of 4 billion years ago. Asteroids crashing into the Earth. As asteroids crash in, they bring new elements that from space that then collide with elements that were here on Earth. And when you have that much heat and pressure, it forces new bonds to be made. And then those new bonds then create new molecules. They can create new organisms. And so you start to have this happening all over Earth. And those organisms start to come together. They start to reproduce with each other. Bacteria can combine together. That's what would happen. The bacteria would engulf each other, and then you get multicellular organisms instead of just a single-celled organism. Because one big bacteria would just go around and eat a couple of them because, well, you're really good at making proteins and you're really good at digesting food. So I'm going to incorporate both of you, and then now I've got all of that in me. And then you start to have multicellular organisms being produced all over the world. And that's why we have such varying life all across the Earth. And you can kind of see that being represented in these different trees of life. The soft chalk and your textbook go more in depth into kind of some of these characteristics of the eukaryotic, archaic, and bacterial domains. So make sure you go through and you're studying that and reviewing that. This is a good point for me to kind of remind you that these videos are supplemental. They are not covering everything. It's just, again, me getting to kind of talk to you about some of this stuff. All right, last little thing here, talking about the scientific method. We'll get into this more into lab too. What you're gonna see with this in 111, what we really like to emphasize are the variables. Being able to identify the different variables within an experiment and being able to kind of evaluate a study or claims even that someone might make on TV. Influencers, I'm big on influencers, y'all. I'm on Instagram as well. I see the stuff I hear what they say, and oof, I have to stop myself from commenting because I don't want to get drawn in. But hopefully kind of looking, we can start to give you these ideas of how you should be looking at information that's being presented to you, how you should be critical of that information, not critical in the terms of constantly arguing and contradicting, but critical in the terms of, well, what are they actually saying? What claims are they making? What are they leaving out? Did they tell me where I can find these resources? Did they tell me where I can find the study? Did they mention any of the methods? Are they giving me percentages, or are they giving me raw data? Because that's something that people love to do, is that they love to either just give you a percentage of something rather than giving you the raw data. And you really can blur lines when you start to play with that. And so the scientific method, it kind of feels played out when I teach this in class in person as well, and I can see people roll their eyes when we talk about the scientific method, because you're used to it from that very much kind of middle school, high school mindset where, okay, yeah, right, it's this that you're seeing right in front of you. You make an observation and you ask a question, and then you start to make a hypothesis. These are all great. What we're going to start to get into are these last parts of it that you probably didn't do as much. You probably don't have as much experience with. Designing experiments, collecting and interpreting data, drawing conclusions, peer reviewing. This is the stuff that we start to get into now. So kind of hitting on the components of experiments and looking at variables. The variables are the aspects of the experiment, right? We have three different types: independent, dependent, and standardized variables. So the independent variable is the thing that changes, right? It is independent of the other things. It can do what it wants. So it is the thing that you're going to be changing within your experiment. The dependent variable depends on the independent, right? It is what's being measured. And then the standardized is what all stays the same. Everything else has to stay the same. So any experiment is going to have these three variables. Depending on the experiment, it might be multivariable, where they actually have more than one independent. That's something that you should not do until you are working with a very seasoned researcher, because it gets very messy when you start to do multivariable stuff. So this one kind of experiment they're showing right here, you kind of have this coffee. And it's like, oh, why does my coffee taste different each day? What makes my coffee taste different, right? This person is kind of curious about that. Well, they decide then that they think that it's the temperature at which they're brewing the actual coffee beans, not the amount. You could have this person could have chosen the time of day that they were brewing the coffee. They could have chosen something like the amount of coffee beans they were choosing. Maybe they used different coffee beans each time and they didn't even think about that. So instead of doing the changing the water, they just changed different beans. Who knows, right? You could choose any kind of aspect of the process. You could choose the different cup, right? Maybe sometimes you drink it out of a glass cup, sometimes you drink it out of a metal cup, and that can change the taste of it. So any kind of variable can be changed, but you want to make sure that you're very clear about what it is that you're changing, and then what it is that you're keeping the same. So you're only changing one thing here, everything else stays the same. So standardize the mass of the beans, the amount of coffee that you're putting in each one, the amount of time that you're letting them brew for, the amount of creamers or milks or sugars that you add in. All of those things would be standardized, so that they don't start to sorry, my brain is blanking on the word that I want to use here. You want to make sure your standardized variables are staying consistent, so that way they don't start to influence your independent variable. They don't start to throw off your experiment, which again is a lot what happens when you start to hear people, especially influencers, making claims about things, products and supplements and vitamins and things like this. A lot of it comes down to bad science, comes down to not being clear about what the variables are, or not being clear about what the conclusions mean. A lot of times that's where science can get kind of messy, because it will make conclusions, and then those conclusions aren't always interpreted correctly. And so if you don't understand the science behind a conclusion, if you don't actually read through the interpretation that scientists provide for that, it's really easy for influencers, for companies, for marketing strategies to just take a claim, take a conclusion, and then make a misleading statement about that, intentional or not. So it really is important that when you're using science, when someone is trying to use science to make a claim, that they provide the study that they're using as their reference, so that way you can go and look at that study and see, are they even interpreting it correctly? Did they read the results correctly? And yeah, are the conclusions correct on that? So it's something that hopefully you can get very interested in with this class. The last two parts, the control group and the experimental group. The control group, that's the one that can be used as your baseline, the thing that you don't change. So while you can have standardized variables across all of your groups, right? A standardized variable, things like the amount of coffee, the weight of the beans, the amount of creamer, those kind of things can be standardized throughout all of them. The control group is the ones that you can see here, it says only been treated with water. Those are the ones that are just your standard non-adjusted. Experimental groups, those are the ones that you can start to change with. Those are the ones that you can start to make experiments on, make changes to, that you would actually then be measuring. Well, you measure all the results, but those are the ones that you're going to be using for your conclusions. So again, for lab, we'll get a little bit more into the scientific method and looking at some questions for that. But hopefully this just was a nice introduction to this first chapter. And again, hopefully you will join me next week.